So, ladies and gentlemen, I'm very glad that you are here and that we can have our Brodmann Symposium. 2018 is the year of the 150th anniversary of Korbinian Brodmann's birth, as well as the 100th anniversary of his death. Given the enduring importance of his work for brain mapping, the dual anniversary seems to be the right time for an OHBM symposium, which aims to remember Brodmann's life and work, but also to demonstrate his influence on and importance for actual research in various fields of modern brain mapping. The first slide, please. Yeah. Brodmann published a series of seminal contributions which represent the anatomical basis of modern brain mapping. His map of the human cortex is still used for localization of MRI data. An actual cited reference search in the Web of Science database resulted in over 4,000 citations. A search within Google Scholar, which includes books, resulted in over 6,600 citations. And the interesting thing is that these citations exploded since the upcoming of functional neuroimaging. Apparently, his work was accepted by this community and cited more and more, and it's still increasing. Next slide. Nowadays, his schematic map of the human cortex seems to be the only still actual part of his work. However, his theoretical concepts of organizational principles and the evolution of the cortex, as well as his discussion of functional implications, are equally important parts of his heritage, though largely unknown to this community and to most communities in our field of neuroscience. This is surprising, because his famous monography from 1909 has been translated into English and published by Lawrence Gary, and it's available. My talk will focus on two things: on the life of Korbinian Brodmann, which is an exemplary life of somebody who has completely changed neuroscience at the turn of the 19th to the 20th century, and which is of enormous importance for mapping. And second, on major concepts and the results of his work. Next slide. Korbinian Brodmann's life was full of setbacks and tragic events, career problems, but also favorable moments. His life and work seems to comprehensively focus the rise of modern neuroscience. The social problem started with his birth as illegitimate son of Sophie Benkler on the 17th of November 1868 in Leggersdorf, a small village in South Germany. His mother, Sophie Benkler, had to work as a maidservant in the house of the wealthy Brodmann family. The mother's family was very poor, and his father, Joseph, did not marry Sophie until Korbinian was 18 years old. From this on, his family name was Brodmann. His Christian name, Korbinian, which is also very unusual in Germany, was given to him by the priest of the village, a usual custom at this time for illegitimate children. After 6 years of basic school at his birthplace Legersdorf, he succeeded to finalize his school education at the Gymnasium in Konstanz in South Germany. After Gymnasium, he studied medicine in Munich and Würzburg, where he attended the lecture on medical physics delivered by Röntgen, the Nobel Prize winner in discovery of the X-rays. Then in Berlin, where he attended lectures not on brain, but on Immanuel Kant's philosophy. And in Freiburg, he finished his studies as medical student with the final exam. The year 1896 this seems not to function. The next slide, yeah. The year 1896 brought the most important encounter for Brodmann's future scientific activities. He met Oskar Vogt, a flamboyant personality and ambitious scientist who had just returned to the Spa Alexanderspad in the Spruce Mountains from his studies with Desjardins in Paris. Vogt convinced the 2 years older Brodmann to specialize in psychiatry and hypnosis. Brodmann spent the following year in Berlin to improve his knowledge in neuropathology and neuroanatomy. In 1898, he moved to Leipzig, met the experimental psychologist Wilhelm Wundt and the psychiatrist Paul Flechsig, who analyzed the appearance of myelinated fiber tracts during ontogeny. During this period in Leipzig, Brodmann also finished his MD thesis on chronic sclerosis of the ependyma. From 1898 to 1900, Brodmann worked in the psychiatric hospital of the University in Jena, where he met Erich Haeckel, the great fighter for Darwin's work on evolution. Haeckel's concept of evolution plays a central role in the future work of Brodmann on the homology of cortical areas in mammals. During 1900 to 1901, he was physician at the Municipal Mental Asylum in Frankfurt, where he met Ludwig Edinger, one of the founders of comparative neuroanatomy, and most importantly for his further career, the neuropathologist Alois Alzheimer. Alzheimer explicitly recommended to Brodmann he should seek his future as a neuroanatomist. Let's briefly come back to the time in Alexanderspad because of its importance for the future of both Brodmann and Oskar Vogt. Alexanderspad was a fashionable spa for upper-class people. Vogt successfully treated as physician and hypnotist the Krupp family, including Friedrich Alfred Krupp and his wife Margarete. From this time on, he became the personal physician of the Krupp family, which owned the worldwide largest industrial complex producing steel, warships, tanks, and cannons. Friedrich Alfred Krupp was the so-called Cannon King. and he had great influence on Emperor Wilhelm II and was instrumental for arming the German troops before World War I. Vogt's relationship with the Krupp family was extremely important for Brodmann's and Vogt's scientific future. Brodmann received a postdoc position in the so-called Neurobiological Central Station in Berlin, which had been founded by Vogt in 1901, a completely privately financed and supported institution financed by the Krupp family, the steel tycoons, and Vogt's income as hypnotist. You see, it's a very unusual way to introduce brain mapping, brain research, because Vogt was never an academician. He was never a professor at the University of Berlin. He was an outsider coming to the university and with the support of the Krupp family and of politicians. Vogt's relationship with the Krupp family was therefore extremely important. In 1902, this Neurobiological Central Station changed its name into Neurobiological Laboratory of the University of Berlin. This was not simply a renaming. It was the beginning of a big battle and a revolution in Berlin's academic structure. Now the suspected private central station was to be transformed into an official department of the University of Berlin. Vogt immediately faced the furious resistance of the majority of the professors in Berlin. Despite all activities of the university, the imperial ministry decided after the intervention of Friedrich Alfred Krupp that the Neurobiological Laboratory of the University of Berlin had to be installed under the leadership of Oskar Vogt. The foundation of the new department had also a positive effect for Brodmann, because he received a position in this department and was for the first time financially secured by the state. The years between 1901 and 1910 were the period during which Brodmann's pioneering work on cytoarchitecture and localization was created. He published the results in 9 original papers and in his famous monography from 1909. The monography is a synthesis of his previous cytoarchitectonic studies and contains the schematic 2D drawing of the human cortical map, which nowadays dominates our view of Brodmann's work. The highly important text of the monography is often not read. Brodmann criticizes any attempt to explain the organization of the cortex by monopolizing the impact of a single aspect of his morphology, such as single cell types or cortical layers. He clarifies this position by conceding that certain cell types have certain functions. So here, the term function does not mean sensory or psychological functions, but basic capacities like inhibition and excitation. When I prepared this talk, I had the chance to see a lot of original, never published documents of Brodmann's work, like this image here, which shows the border between V1 on the left side and V2 on the right side, and the handwritten remark by Brodmann, which means human calcarinotype with transition to another area. So this was the beginning of a cytoarchitecture based on comparative neuroanatomy. Further, Brodmann states the polymorphism of cells, the strict regional separation of certain cell types, and the regular appearance of similar homologous cell types in identical positions on the cortical surface in all mammals, all that justifies the view that in the cerebral cortex a broad division of function has occurred. Central to Brodmann's concept of parcellation and localization are two questions. What is the principle of cortical organization in all mammals? And what is the cortical area and its relation to brain function? He described the general consequence of his work as follows. The most important finding of our comparative topography of the cerebral cortex is that its cellular organization manifests a common architectonic plan in all mammals, a common pattern of layers. First, we were able to distinguish two types, the heterogenetical and the homogenetical types. Further, we ascertained that in all mammals, the homogenetic cortex manifests a regular series of variations, types, that themselves all stem from a basic histological form, the original six-layered type. He continues, finally, we were able to show that thanks to these homologous structural formations, it is possible to demonstrate an essentially standardized organization of the surface of the hemispheres throughout the whole mammalian class into spatially delineated zones or cortical organs that we dominate regions or cortical fields. Nowadays, the heterogenetic type is the allocortex, divided into paleo and archicortex. The homogenetic type is the iso or neocortex, subdivided into numerous areas. What supports Brodmann's position that this statement holds true for all mammals? It is stunning work. He studied at the beginning the histological quality of the different regions of the neocortex in non-human primates, in a Sarcopithecus monkey, and yellow and demarcates the primary visual cortex at the occipital end. Yellow, more in the frontal lobe, depicts the premotor cortex. Red, the primary motor cortex, and blue, the allocortex or periallocortex, the old part of the brain. And you see that these areas occur at precisely the same positions in human and in monkey brain. There is also a study of Brodmann in a lot of very exotic species. For instance, Coleptus kinkachu, a bear-like animal, or in Erinaceus, which is the hedgehog. And in all these animals, he found again the basic structure of the motor, the visual, the premotor, and the allocortex, and its topological relations between these areas are more or less the same in all animals. So what Brodmann said was an area, a region, is not only characterized by the cytoarchitecture, but also by its homology with other mammalian brains. He expanded his comparative studies, and these are very, very rare handwritten drawings by Brodmann, which I could get from the Brodmann Museum in the little village where he was born. And you see here lateral views of a hemisphere of platypus. On the left, a monotrema, and a spiny anteater, again echidna, also monotrema. And Brodmann did not just map a brain separately from all other brains. He permanently compared the different species and tried to find exactly the same cytoarchitecture in the different species to characterize an area as an area of the cerebral cortex in his sense. He studied also marsupials, for instance here a kangaroo brain, and red is the primary visual cortex, yellow the olfactory cortex. And here the guenon, it's a monkey, his favorite model except the human brain. And you see again that he labels the different areas with symbols, which are also found in his human brain map, indicating the homologies between the areas. And this is a very early hand drawing of Brodmann, where he started his work on the human brain. It was a horizontal section through a human hemisphere, and his inscriptions, when you look closer to it, describe things like layer 3 is white and has large pyramidal cells, layer 5 has smaller pyramidal cells in this area, but not in the motor cortex, and so on. His answer to the second question, what is the function of an area, is still interesting. Brodmann wrote, only functions of the most elementary kind can be associated with histological elements, but never complex psychic images. Then he introduces the network concept by saying, we must therefore reject a quite impossible concept, the idea that an intellectual faculty or mental event or any other complex higher psychic function should be represented in a single circumscribed cortical zone or area, which one calls this an association center or anything else. One should envisage the situation that in each particular case supposed elementary functional locus are active in different numbers, in different degrees, and in different combinations. Thus, Brodmann was neither a naive localizationist nor a late phrenologist. In 1908, Brodmann submitted his habilitation thesis on cytoarchitectonic parcellations of the cerebral cortex in prosimians. The thesis, a requirement for obtaining a professorship position in Germany, was officially rejected by the University of Berlin because of the battle between Vogt and the university when founding the Neurobiological Institute, and this animosity between Vogt and members of the medical faculty hurted the career of Brodmann and his work very much. As a consequence of the dirty battle on Brodmann's habilitation, the atmosphere between Brodmann and Vogt worsened. Finally, Brodmann left Vogt and Berlin and moved to the Clinic for Psychiatry and Neurology of the University of Tübingen. His habilitation was accepted without any change by the University of Tübingen in 1911, and he became an appointed professor. Between 1910 and 1914, he published several summaries of his previous work together with anatomical and physiological descriptions of the brain. However, the beginning of the First World War stopped all his scientific plans. Between 1914 and 1916, he had to serve as a physician in a hospital for soldiers. In 1916, he moved as prosector to the Niedleben Mental Asylum near Halle in the eastern part of Germany, where he stayed until 1918. From the first time, he was assured by this position of a reasonable material security. In 1918, he reached the climax of his professional career. He was appointed as head of the Department for Topological Anatomy in the famous Research Institute for Neurology in Munich, which was founded and headed by the eminent psychiatrist Emil Kraepelin. The neuropathologist Alois Alzheimer and the anatomist Franz Nissl were renowned heads of other departments in this institute. However, Brodmann's fate annihilated all great brains. On the 17th of August 1918, he developed what seemed to be a simple influenza, but after a few days, signs of septicemia appeared. It is thought that an old infection that he had contracted during an autopsy sometime earlier had flared up. Brodmann saw his illness as a way of catching up a backlog of work. He seemed not to suspect that this was not to be. One day, he was seen to be making writing motions on his bed with his fingers before sinking back dead. Brodmann's work remains a seminal landmark for localization of cortical areas, and he can be seen as the founder of anatomical brain mapping. Long before the time of neuroimaging, neurochemistry, and cell biology. During the first decade of the 20th century, he paved the way for brain mapping. He is the tragic giant on whose shoulders we stand. Thank you very much.